So I'll be talking about two components of other programs and analysis tools that we have at the NIH Center that we find useful for people working in the area of computational biophysics. And I think some of you have already tried multi-seq before. And let me just give you a quick background to it. I think all of you know that on the structures and the sequences that we work on today, they're all a result of evolution. So it really makes sense to use evolutionary concepts in analyzing even your molecular dynamics trajectories, or even earlier as you set up the system, and don't get too in love with a particular interaction if it only happens to be there maybe in one species, but it's something else in 1,000 or hundreds of thousands of other sequences. So what I'll tell you about is the multi-seq and the evolutionary tools that we have in it. We were very fortunate. We worked with the time we developed it, the leading evolutionary biologist. His name is Carl Woese. He got the Crayford Nobel Prize in biology for the discovery of this third domain of life, which is now in all your textbooks, right? And so we tried to mimic how he would handle sequences and did the comparison between these three domains of life to be coming up with this phylogenetic tree. So there are going to be the standard tools of bioinformatics in multi-seq. It's going to compare sequences, compare structures. It'll map multiple sequences onto structures, but it will also have some high-level bioinformatic algorithms that Carl said, and this is a very nice quote of his. He says, VMD is far from a simple visualization tool for biologists. It's a true thinking tool. And without it, a whole class of biological hypotheses simply would not exist. And some of those high-level tools are things like he did, like he looked at signatures in the ribosomal RNA. So he made this tree by studying the 16S rRNA. So you have to do it for hundreds of genomes. And then you look for signatures that tell you that that's a eukaryotic system, archaea, or bacteria. And then what we've done is gone from having too little information when he first started to too much. So you really want to have evolutionary profiles for both the sequences and the structure, somehow some representative set that represents the diversity that is out there. So that, as I said, that you don't get too in love with a particular interaction, unless, of course, you're trying to do a design problem. And that's fine, too. The tools are set up. You don't have to know evolution. It helps, though. All right, so if we're going to do evolution and comparative analysis, we need to have multiple sequences. And so what are we going to do with those multiple sequence alignments? Well, we're going to look for patterns of conservation. So here, blue means something is completely conserved. Gaps, I think I don't need to explain. And so what does that tell you? Besides patterns of conservation, it tells you where the active sites are. Those conserved residues could be also important for folding or some other function of the system. Then there's always gets to be a question, how many of these sequences should be included? And again, I'm going to say that that's going to depend on what you're trying to do. If you're interested in working mostly on bacterial systems, then you're wanting to look at it, bacterial systems. This is a list of eukaryotic sequences. So if you're working in the other domain of life, you'll probably always be comparing the human to the mouse and to yeast. And you see some things that are in yeast are not in the mouse or not in the human. So it's good to keep this in mind, as I said, when you're using any of these as model systems. So now the next question is, where do I find these sequences and structures? Well, that has gotten a little bit better. The majority of them are, well, they're all over the world now. Every continent has their set of experts, but they try to mirror everything in our national database. In Washington, it's called NCBI. But if you want the last word in something, you need to know who's working on that, and oftentimes at their particular site, they'll have more sequences and more structures that you can have access to. Then the next question is, how did I get this alignment? Now, some questions are so ubiquitous that now they're even beginning to publish multiple sequence alignments. And then if you come in with something new, a new structure, a new sequence, you just align it to the given set. But that's not true for every case. So in fact, the majority of cases, so you have to be prepared to make this multiple sequence alignment. So that's also in multi-seq. The most precise alignments that you can get are done pairwise. And then with multiple sequence alignments, they're almost all iterative. And almost every 2 or 3 years when I give this part of the lecture, there's someone sitting there who has developed one of the new multiple sequence alignment methods. He or she will complain, why don't you have my method in there? We're happy to do it, and I'll show you one that we did put in. We've even updated it, but it depends on you guys. You have to make the code available. You have to allow the executable to come into this program. We have, as I said, some of our own algorithms, but if we incorporate something from you guys, it has to be available to the large public. So where have been applications of these sort of evolutionary analysis? Well, certainly in looking at an understanding of folding, which you see on the right, or assembly, excuse me, on the left, and assembly that you see on the right, and the energetics involved with both of those processes. So this is a picture of a protein folding funnel that I developed with my 2 colleagues, Peter Walnes and Jose Onacek, oh my God, almost 20 years ago, when we were getting the whole field started into using simple models for protein folding. So then now you could look at just particular structures if you want to understand their functions. But at that time we were concerned about the dynamics of the folding or the assembly. So when we first came out with the multi-seq in 2006, it had these basic tools in it. You could bring in sequences of proteins and RNA. You could bring in structures. You could take a multiple sequence, multiple structure alignment, so there's very little variation, and then map the sequence information onto the structures. It was important that you understood something about those sequences, so we had to bring in a lot of metadata information, because at that time really the databases for sequences didn't talk very well to structural databases. So there was a list that said this sequence does have a structure, it's sitting over here in the PDB database, and it belongs to this domain of life. So when you're bringing in that metadynamic information, you're bringing in all this crosstalk. And it used to be sort of manageable. It's getting larger and larger. But for the purposes of this workshop, we've given you the reduced view, reduced set of files, just so you get a flavor for it. Otherwise, it'll take, you have much bigger files that you'd have to download to be able to use it. So now let's say you take these sequences and you want to be like Carl Vose. How related are they to one another? Can I take maybe the rRNA again and check whether his signatures are there, or I take a protein and I do it? You're going to get these trees, and now the tree is just sitting on its side here, and you see, oh my God, there's a lot. That's almost, I can't see the forest for the trees. Can I eliminate some of those? So we have an algorithm that helps us get non-redundant representative sets. It's called a QR algorithm. And then finally, if there are new sequences out there, you might want to bring them in through BLAST. I remind everybody, BLAST alignments are just an approximation. You have to go back and do something much more careful with them. And that means you're going to have to edit the alignments. So you have to not only read them in and view them, you have to be able to edit it. All that is in multi-seq. And then in the more recent editions of multi-seq, we introduced some of those, I said, some of those high-level abilities to look at signatures. As you look at more and more the genome, you better develop the feature called zoom, or you can't, even see everything on a screen. And the trees got more complicated. We also wanted to see where those proteins or RNA, where the operon is sitting in the genome. And then it became really clear, I don't want to sit there and do the same thing. If I want to, compare all the ribosomal proteins, for example, I better start writing some scripts. And so just like VMD has scripting, I think you guys may have done one exercise with that already. There are scripts also for multi-seq. So once you've got sort of a routine in, things that you want to do, a pathway to analysis, then you write it out. And then we have a bunch of tutorials that are down here, and they keep being expanded, revised, They'll be primarily on translation, and again, that just reflects the fact that we developed multi-seq with Carl Woese, and that pattern that you saw, those 3 domains of life, almost every molecule involved in translation and transcription follow that pattern. So we take that as our reference and everything that we further develop. Okay, so what are those proteins involved in translation? They're, as I said, protein RNA complexes, and we've done dynamics on every one of them. So there are these proteins, they're called the synthetases. This one is an amino-acyl tRNA synthetase for aspartic acid. The job of these guys is to put an amino acid onto the tRNA carrying the anticodon for that amino acid. And we've color-coded it here by a structural conservation over all bacteria. So it sets the genetic code. And then once the tRNA is charged, it gets transported to the ribosome by the EFTU protein here. And again, we colored it by conservation of sequence at the interface to the tRNA over all 3 domains of life. There's only normally one version of this in a cell, but it's the most highly abundant protein that's in there. It's carrying all those tRNAs to the ribosome. And these little space-filling units here are shown to remind you that when you do nucleic acids, there aren't just 4, there are 110, because there are so many modifications. If an interaction is important to your simulations, like here, perhaps at the interface to this EFTU, you need to put it in. Right? And then, as I said, it brings it to the ribosome. Here I'm only just showing you the large subunit. Here's the messenger, and here are the 3 tRNAs reading the messenger. So this tRNA got smaller and smaller, and now it's really teeny tiny here. So one has done simulations, as I said, on all steps of this. But now if you look into the cell, you don't see one ribosome. In fact, I asked Carl, how many ribosomes do you think there are there? And even though he got his Nobel Prize comparing the rRNA on the small subunit of this, he goes, 10? I go, ha! Thousands, right? And then you can't use the MD methods that you have developed and used so far to be able to look at the translation and the full pathway if you're going to do it inside of a cell. So that's the motivation for the second part of this morning's lectures, where we start looking at these processes as they function inside of the cell. How can you do it? What approximations can you make? Okay, so quick review. Those guys set the genetic code, and as I said, we've been working on this since about 2003. I think this is one of the most cited papers that's in this field of evolution of structure. We looked at dynamics involving the tRNA interacting with the EFTU. If you do work on nucleic acids, the magnesiums are a real problem. You have to know where to position them. This could be a whole discussion just about those. And then in the motions that you can see and how those motions get affected, I don't think well, maybe the movie works. So one of these movies is at low concentration, and you'll see there's a lot more motion in the anticodon binding arm. But if you have a lot of magnesiums, the base pairing stays pretty much intact. So flexibility is deeply influenced by your simulation conditions. And over here, as I said, we reviewed Carl's signatures both for the large and small subunits to see if they held up after now having over 100,000 sequences available. The short answer is yes, they do. And then we went on to look at what those signatures, what their function in the system, and our latest paper on this, where we compare it with experimental work, was in Nature. So there's been a lot of work just from my own group, but if you go out, and of course, there are 3 people who got the Nobel Prize for the ribosome a couple of years ago. So their papers are the seminal papers in that whole field. Okay, so what's the basic principle behind evolutionary analysis for proteins as well as RNA? Both of them are molecules that result from evolution. So I think we talked about you got to have sequences. The alignments may not be perfect, so you have to perhaps edit them. You would want to to do the evolution, you're going to have to put them in a tree. So we have fast trees to do, full trees. Oh my God, you better do them offline on a workstation. But you can bring back in the tree and then view it in VMD. But the best one, they want, if you're going to publish it, you have to give support values at each of the branch points. Also, you're interested in, did this molecule that I'm studying, evolutionary, did it follow the canonical pattern that Carl built for the rRNA? Or has there been horizontal or lateral gene transfer? You can use multi-seq to also look at those questions. And then finally, where does the product of that gene sit in the genome? And again, as you get more and more information, you're going to want to simplify and carry a representative set along with you. Those are these profiles, right? And as I said, each one of those you could really delve into much greater detail about them, but you'd have to want to do it. And then I would say we have a lot of literature that's connected to them. We try to give the most recent citation, but if you are interested that you should, and don't know anything about it, you should start with the simplest articles that we have, like, it's like phylogenetic trees for dummies type of articles, and then build your way up, because there's so many parameters and models of evolutionary change that can be used in handling those multiple sequence alignments. All right, so how do we get the alignments if you're going to have multiple sequences? The classic method that's been out there for years is called Clustal, Clustal W. They do now have an improved version. This old one used to be really slow. Oh, I covered it up. It took 5 minutes to align just these 200 sequences of EFTU. That's a long time. And so we installed then MAFTI, and because the developer of MAFTI attended the workshop about 6 years ago, I think it was like 2008, came by. He now has updated it, and I think we have this newest version in there, and they ask you to cite this one if you make extensive use of it. We have always some acknowledgement that comes out of the third-party algorithms that we use in our programs. This is very fast. It's like 30 seconds, 5 minutes, right, to do the same number of sequences. And I also felt that, at least compared to the classic W, it gave better alignments. The gaps made a little bit more sense here. We don't have the newest Omega Clustal in there, so perhaps they've improved that too. Okay, so what's important, you'll see later, are the names that come in here. I like to have it also put in some grouping that's all flexible. You can give a short name if you want. So when you're downloading that metadynamic data, that comes in like the official name, the EC number, the common name, the organism, but you can make up subheadings yourself, but be sure to save it. Okay, now that's a multiple sequence alignment. To do the sequence alignments, the most exacting one is to do it pairwise. And again, these are methods that were developed by computer scientists. Perhaps the most famous one is known as the Needleman-Wunsch. It gives you a global alignment of two sequences. So if you have a sequence A and a sequence C, how should I align these things to one another? And if you just think of them as letters and spaces, you could have this many possible alignments. That's terrible. So you obviously want to introduce some metrics into this. So one of the metrics is, well, I'll have a penalty for putting in too many gaps. Sounds reasonable. So if something's really not related to another one, there should be a lot of gaps, and you should see that in the overall score. The other thing is, how related is C1 to A1? You use that and encode that in a thing called the substitution matrix that takes all the standard amino acids and gives similarity scores to them. Again, there are several versions of this, and it really depends if you're trying to align things that you know are very distantly related, then you'd want to use one that has been trained on things that have been distantly related, and the scores are representative of that. If you know that you're working with sequences that should be closely related, then you might want to use what they call like a PAM 80% sequence identity. So those are things that you'd have to decide. Now, I assume most of you have run sequence alignments. Is there anybody who has not run one? Okay, there's only a so, and normally you're just asked what substitution matrix you want to use, but one should really be paying attention to this. So I think maybe this whole part is actually, so many of you have done it, perhaps I can make this part even shorter, right? Because I think you all realize the power of these sequence alignments. And what I would say is the MAFTI is one of the better ones. There's other ones called like Muscle and T-Coffee. They're also very good that are out there. You can bring in any alignment and view it in multi-seq, but these are the two that we offer, this Clustal, the classic Clustal and the MAFTI. So this algorithm, it's always good if you're going to use these methods that you know what the limitations are with them. So in this case, when you're doing this alignment, the algorithm says you calculate a score and you take whatever move gives you the maximum value. So what you could do here is if you've aligned up to I and J and want to continue to aligning the next one, you look up its score over in this matrix, and that's the value of this top line. The other two lines indicate whether you put an insertion and deletion into one of the sequences. And what you can do then is fill out a matrix to do this alignment. So here are like two short sequences. If you fill it out first at the top, so right now it's filled out with the similarity scores from that matrix. So you would look up when you align an M to an M, what's the score? When I align a G to an M, what's the score? And then you fill in the top with the gap penalty. So let's say you had a gap penalty of 6 for one gap, 12 for two, so on and so forth. And then you just start working your way through that matrix. And each time you just take the step that gives you the highest score, right? So pretty soon then you're down here, and that's your overall score for the whole alignment procedure. And then you have to work your way back. How did I get there? Which of those many steps did I take? And then you look at your alignment. So the M to the M, that's pretty clear. G to the G. You could have perhaps aligned this K to either one of these. So there is some redundancy even in the alignments. But the way most of these programs work, and you have to be really aware of this, that even if they're degenerate answers, it's the last one in is the first one out. So that's the score and that's the alignment that you're going to get. Almost no program out there will allow you to give multiple alignments and suboptimal alignments that are coming out of it. Okay, that's just for sequences. Aligning structures is much more difficult, but in principle, you're using the same technique. You start off with some pairwise alignments, then you try to do it iteratively, adding more structures to them, and you have certain scores that are in there. The method that we have in multi-seq is called STAMP. It was developed first to align protein structures, and it was developed by Russell and Barton. And this was their original paper. We took it and then modified it to also be able to align nucleic acid structures. So you get these scores. So there'll be a score that'll talk about the distance between these residues in two different structures, and then plus maybe some correction for loops to allow for conformational changes. And then the total score is made up of these, the length of the alignment, these distance scores, and you do this until you get what you want is convergence. You had a question?
How do you deal with gaps in the structural alignment?
Excuse me?
How do you deal with gaps in the
I'm just going to tell you. Yeah, that's a big problem. Do it this way. First, let me show you the results of it. So they have in their algorithm a score for the lengths of the gaps, right? So we let STAMP do the alignment, also decide on the gap. But because we didn't want to change too much of this algorithm, because it had already been through, due diligence and people had analyzed it. What we do is then take the resulting alignment and look at it and analyze the gaps ourselves. And based on that, we did go in and modify things a bit, but there are different scores that you could be using, particularly when you're comparing two structures. So I think you may have seen it yourself. Like, is RMSD always the best thing to use between two structures? Probably not. Particularly from an evolutionary perspective, even conformations that are in PDB database, a lot of it can depend on what cofactor was, co-crystallized with it. Or maybe a feature has come in, so you have a helix, and this helix is closer to the body of the protein in one structure, and it's further away in another structure, right? But all the local alignments are great. This would have terrible RMSD, but you would get a local score, it would be very good, and then you'd get all the distant relationships. It would have greater error to them. So RMSD is not great, and then because we worked on protein folding, many people wanted different scores to represent how close one was to the correct answer. In fact, at one time there was a joke where you're not good until you can reproduce the artifacts in the crystal structure, right? And there's something to be said for that. Okay, so what we were interested in is, can we use STAMP to structurally compare any of those assemblies used in translation? And the most toughest one is, of course, the ribosome, right? And this is just a large subunit with, say, two of the some 30 proteins on it. One's from E. coli, a bacteria, and one is from Marasmortia, which is an archaeal system. And then you overlap them, and then you report the alignment in multiseq. So this is the 23S, that's the RNA that's in the large subunit. And there's only like one or two places where they vary at this part of it. And then the two proteins, there are actually more differences between the L2 in the archae and the L2 in the E. coli, right? But as far as I know, there's no other program except ours that can do this for something as large as the whole ribosome. So that was, we needed it, so we had to develop and dedicate some time for it. So I'll get to your gap question in just a second, but let me go back now and just sort of summarize sort of this evolutionary approach. So we have Carl's reference tree that was done on rRNA. Here are the bacteria, E. coli. Everything, these are the darlings of people expressing proteins. They don't care if they have proteins from this or this domain. They express them in E. coli from that domain. You have archaes, which are becoming increasingly important because you find them in the human microbiome, as well as in geysers and maybe on Mars even. And then, of course, we're sitting over here. I didn't even put us there among the plants and the fungi. And all right. So now if you look at this and turn this tree sideways, this is like looking at one of those synthetases that loads leucine, the amino acid leucine, onto the tRNA, that loads leucine onto the tRNA with the codon for leucine. And you see immediately one of the problems, this is from a paper that Carl published back in 2000 with Dieter Sohl from Yale, is that you always have many more bacterial sequences. They're easier to get at. These are much harder. You remember we needed this whole initiative in the human genome to even get that jump started. Now you have, you have plants, you have mouse, you have cow. This is getting much bigger. But if you try to do a representative, we're looking at multiple sequence alignment and didn't pay attention that you have 3 domains of life, you would overload any statistics you do with bacterial inferences. And that wouldn't be proper unless you wanted just to work in the bacterial systems and you're interested in that. So keep this in mind. The databases are biased, and they're biased by the ease with which you can get sequences and structures. All right, so one of the things you have to watch out for is life is not always that simple. As I said, the proteins and the RNA involved in translation and transcription do, to the most part, follow those patterns, but you occasionally do have horizontal gene transfer, where suddenly the bacterial signature is sitting there in the eukaryotic system, right? So it's like you've got this tree of life, and then there's some little crossings. It's like somebody went and then grafted something from one domain of life into the other, right? So you can look for that, and even in those systems involved in translation, there are a few examples. One, I don't want to get into the evolutionary theories about it, but sometimes they talk about the early and late amino acids. So the leucine that we looked at before, it follows the canonical pattern, but you also have a basal canonical pattern where you only have archaeal and bacterial, and now the eukaryotics are sitting here in the bacterial system. And then you can have one that's called non-canonical, and that's for cysteine, cystinoyl tRNA synthetase. And there is actually more pattern in there than you think, because it's just like certain bacteria have given their copy off to eukaryotes that it actually maybe coexisted with. So even in this pattern, there is a pattern. But right now we would just call it non-canonical. All right, now to your question of the gap. So now if you looked at the structures and you wanted to compare these two, and this was a place where they disagreed, for example, well, you'd want to have a score that's based on how much is aligned, and then a metric for that gap. And so you're going to use distances. Compare the distance, say, between I and J on one structure with the I and J prime on another structure. And you'll be more tolerant the further away I and J are from one another. Right? So that's your score. And then just to give you a feel for, let's grow that gap in. And you would have a Q score. If there's no gap, that looks pretty good alignment. That would be a score of 0.82. The next one, you're starting to grow the gap, 0.7. And then that bigger loop would be 0.62. So then if you took all the structures that were for, say, the class 1 synthetases, those things that set the genetic code, and you did and first just looked at the aligned parts, and then you looked at the aligned parts plus the gap, you see what this QH metric does is it moves everything down so that really all those synthetases that do similar activity have a Q score of about 0.4, right? So just so one gets a feel for the variation that you can have. Now let's compare a sequence tree with the structure tree, because structure also encodes information. And this was the tree that sequences that Carl and his colleagues did. So you have, they didn't have too many of the archaeas, of the bacterial systems, so it's sort of up here. But you'll notice there are 2 copies of this one Deinococcus, because there's one that has a clear bacterial signature and one that has an archaeal signature. And now, again, we don't have all those structures available to us for these sequences, but we have enough. And they were so interested in this that they went and got the structure for these 2. And indeed, there is structurally a large difference in those. And let's look at that difference. So if you look at the system sort of representing all the archaeal branch up here, they have more or less, and you overlap them and do structural conservation. So blue is really structurally conserved. Red, a bigger loop in one than another. And now let's go over and look at the guys in the bacterial system here, right? And there's this huge insert that has come in, right? So it is a definite difference has developed as you took this protein from one domain and had it in, and it developed into in another domain of life. But it does the exact same function. This loads aspartic acid onto the tRNA with the aspartic acid codon. I'll skip this just to show you sort of where at what level of subclass or speciation should you be using more sequences and structure information. I think it's more important to remind ourselves one of the more difficult problems when you're working with sequences, and this used to be more so when I think protein structure prediction was such a hot field. I think it has really cooled off a lot. But one of the lessons that we learned there is there can be multiple proteins that do exactly the same function, but they have totally different active site geometries, or even just folds in general. So if you look into the core here, these are parallel beta sheets. These are anti-parallel. This one goes this way, and this one goes that way. But they both load lysine onto the tRNA. And there's some for lysine, and there's some organisms that have both of them, and there's some organisms that have only one, and others that have only the class 2, right? And when you say class 1 and class 2 synthetases, they're always referring to this inside active site geometry, because the parts that interact with the anticodon will vary more frequently. The last word about using structures, using sequences, is really the sequences do define a more recent evolutionary events, but whenever you're using things from the PDB, you've got to be careful. The conformational changes can be as great as changing the structure between two species. So these are two of the same proteins. One is just with an analog and one is without a cofactor. And if you look at the structural comparison, it's 0.8, pretty high. The sequence identity is exactly 100%. Go over here and look at the, I think, ones between two different arches, and the Q score is 0.89. It's even higher than over here, but the sequence identity is only 7%, 70%, right? Now, sometimes you look at this and you go, okay, main reason to be aware of this is oftentimes when you're dealing with structures, there's some part missing. And you have a sequence of it, but how am I going to model that thing in? There are programs that do it, but you can also have a good idea whether you've done it correctly yourself by just going and look if that loop is conserved, if there's any structure that was obtained maybe through NMR that had it in there, then I have a few guesses to be working with it. You don't always have to rely on programs like Modeler that are out there. They do a good job for you, but you need to also have independent validation. So last word on this, let's just follow the evolution, say, of this synthetase that loaded alanine, say, into the one that loaded aspartic acid. And so again, primarily it looks like it has those canonical structures, but there is a slight variation as you go to a different synthetase. And now if we compare today's aspartic acid with this one, that's where the signal is sitting, and then you represent it down here in terms of sequence identity and structural identity, and that yellow dot is sitting right here, so structurally they're pretty high, but sequence is it's less than 10% sequence identity. But you could use that structure to model the other one if you want, even though the sequence identity is only 10%, right? Okay. Next thing that we have to watch out for is, as I said, we have really gone from a state of having too few to almost too much information. So if you aligned all the synthetases, say for class 1, oh, this is what you'd get, and you can hardly read the numbers. So you want something that goes in and selects a representative out of each one of these and tries to do it in the sense that they are most linearly independent from one another. So all we've done is either encode the structure or the sequence in a matrix. So this is this QR algorithm. It was actually developed by Caltech engineers for earthquake analysis. But you have the proteins listed here. You have the alignment in this direction. And in that direction, you have either the XYZ coordinate and some measure of the gap there, or if you have sequences, that'll be 20 dimensions in that way. And then this algorithm just keeps reshuffling the proteins, looking at how different is protein 1 to protein 2, 2 to 3, so on and so forth. And then the first ones then will give you the subset that are most representative. So it does it for you automatically. And this is just summarize what I told you. Either in, if you're doing structure, you have a 4 space. If you have sequence, you have a 24 space. And this is, if you're interested, there was several articles on how one can use this to find an independent set. Okay, there's one adjustable parameter. I don't think we even allow you to change it. We can tell you where it is, and you can go do it yourself, but I wouldn't do it because that's what some of those papers were about, is trying out a whole bunch of different proteins, deciding what's a good adjustable parameter, and then we called it a day and left it there, right? So far, nobody's complained about that, all right? Now, again, now let's just summarize this part of the lecture, and let's look at the evolution of structure and function of that synthetase that loads aspartic acid. Let's start out, we have our minimal tree here. Notice that we start off here with the one for aspartic acid, but now we're getting into serine, proline, lysine. We're moving up the tree. So now if we look what all those have in common, the only thing they have in common is that core domain I just showed you a few slides ago. Let's go down to this branch point. Oh, now they all have added this anticodon domain, right, that interacts with the bottom part of the tRNA, where the anticodons are sitting. And now let's keep going. Let's get closer and closer to the one for bacteria. Looks pretty much the same. The one up here, this would have been good for either the aspartic acid one or for the lysine system. And now let's zoom in. Let's get closer. This is sort of for all bacteria. At this point, this was the structure that would have been for both arches and the bacterial systems. And then if I move to that last point, that's only the bacterial branch, and so now you have the bacterial insert in there. So when people are doing design, this is how they use multi-seq. In fact, colleagues like Schultz and Li Wang that used to be at Scripps, they did this, used this tool to try to expand the genetic code, to bring in new amino acids. And if these are the basic molecules you have to work with, they hijacked one of the synthetases, modified some things in the active site, and then also modified some of the tRNAs. And this is how you could check what has been changing as you moved along in evolution. Okay, so as I said, structures, there will be less variation, at least in the core part. The sequence variation will be enormous, right? And so it's always good to try and look at various levels of evolution, either the family or superfamily or things more distantly related. And exactly how many structures and sequences you include, I think you'll have an intuitive feel for it, but you can always run our QR algorithm, which tries to give you at least an independent, it tries to eliminate redundancies. Because most time when people say non-redundant database, all they've done is taken out things that are 100% similar. But everything else is like 99% similar to it. So you really want to look at the diversity that's there. So I think now you can understand a bit more what's in this. Is this thing working? Yes. What's in these tools. But I'll just give you a blow up of a few of them. So as I said, you can combine sequence and structure. So this part here, we've aligned all these structures and of just the proteins. And then we went and we looked at the sequences and then mapped that on to the structures. Okay? The structure alignment itself will not have as big a color change as the sequences will. And again, when you have complexes, you have a choice. Do I want to align the protein or do I want to align the RNA? I would suggest you try both because you can pick up subtle changes in the interfaces between any two complexes. If you want to do a full evolutionary study, as we did in developing multi-seq, when we did it in 2008, this was together with Carl, we could bring in all these sequences and all the RNA sequences. There are like 100,000 of the RNA and about 400,000 of the proteins and the ribosomal proteins. But if you go and look at, say, green genes today, it has, as of 2013, 1.2 million sequences. So I don't know if I would do that on a laptop, or I would think about how to simplify it. So you can, of course, do it on a workstation. There's no problem whatsoever. But to do it on a laptop, you would probably want to use a smaller version of it. And that's why the one that we carry around, like for the workshops, is pretty representative of it. We just want to be sure that everything that we're interested in the tutorials is in there. And so you might have to modify it as well. As you get to looking at bigger and more sequences and more of the genome, as I said, you're going to really want to make use of two things, the sequence editor and the zoom feature that you see here. Just zoom out and see where there seems to be a big shift somewhere, and then go back in and zoom in on that part and study it. You make the trees. Now that you have all this metadata in there, you're going to want to color this tree. Like this would be something that you would just pull out a blast. There's a bunch of numbers for those sequences. You could have it at least tell you what domain of life they are. It could give you a number if you had the EC number there, and then color it by the domains of life. Very useful tool. To get those support numbers, you really should run it through something like RAXML used to be for many years the best maximum likelihood tree analysis tool. But you do have to run that on a workstation. Do not try to run it on your laptop. We have the instructions in the back of Multiseq how to run it, but don't do it. It's just too slow. Okay? Instead, what we do have is something called Phylip that goes really fast. It's not as good, but it'll give you the general character. And then the next thing is, just like with VMD, you're going to want to get used to scripting. If you, like when we were studying the ribosomes, and we're doing it from like 100,000 different species, we're not going to sit there doing this 100,000 times, right? So you write a script, and to do the RNA, we wrote another script to do the ribosomal proteins. And it also helps you discover if you're making any mistakes a bit more quickly, because it's really totally reproducible. The other interesting scripts that we have written, and I think some of them are in the Multiseq tutorial, like let's look at, say, this ribosomal proteins. At least with the bacteria and the archaeas, many things are in operons. So, and that's doubly, one of the most conserved operons are the ribosomal proteins and the ribosomal RNA. So let's go find that operon. So you're looking where its location is in the genome. And let's say I want to make a tree, but let's just do it first on S4. That's one of the proteins that nucleates the small subunit. But let's bring in all the neighboring information. How conserved is that whole bloody operon? So you write some code that brings in the genomic sequence, and then so many nucleotides to the left and to the right of that. Make a tree, but make a tree just on the one protein there, that S4 that I put an arrow by. And of course, life is never that easy, right? So S4 in the genes is known as RPSD. So that's why you also lead that list that tells you, oh, that's just another synonym for what the protein chemist call S4. And the geneticist calls RPSD, right? So that's what we're now looking at, and then you can go color it. And that operon is really quite conserved. It's normally S4 sitting here, and these are usually its neighbors. There are some variations, so we looked at them because sometimes they have two copies of it, or S4 has moved over someplace else in the genome. Mm-hmm. Excuse me?
Why is the polymerase opposite of
The polymerase. Because it's really important, right? Excuse me? Okay. Let's go back here. I only looked at this much of it. So in this particular operon, this is called so-called alpha operon. this is there. I don't think I have, so since you know a little bit more, how about sec-Y? Do you know what that does? Okay, that exports proteins into membranes, right? So, and then in another one of the operons for translation, you'll find the EFTU, and you'll find the other subunits. So I don't know why it's got split up. Maybe that is the most conserved unit of it. It's probably somebody who knows something more about the polymerases knows this. But the other subunits, they're connected to these other regions, these other operons, right? All right, so we were interested, and for us, we were studying primarily where the ribosomal proteins were. All the ones involved in the small subunit have an S with them, so we're seeing S4, S11. It'll come up again because now we've made a whole model of translation in the cell, where we start from the operons, transcribe it, make the proteins, and assemble the ribosomes. So one of the other things that for us that was interesting is that there's a second copy in this Clostridium. And it turns out many organisms that have spores, make spores. In the spores is the ribosomal assembly machinery there. And they'll have a second copy sitting there. It's not as neatly laid out as in the main copy, but these are the kinds of things that you see. Doing this map, this you had to go outside of VMD and multiseq. So you had this picture coming out of multiseq and writing the script. But then to color it, you go into something like Illustrator. Now, this is actually a really good idea. This we did back in 2009, published this. But some colleagues at the U of I have now taken the pan-genomes for many microbes, like they've done it for the methanogens and the Clostridia. They had a grant to do it. So it's already been pre-compiled, and you can just pull out these comparisons and look at them, which is pretty cool. So this is what a lot of databases are trying to do now. If you go to DOE's, JGI, or any of the microbial genome databases, they try to give you some context about genome content, right? It's just that you can't manipulate it so well. That's why we wrote this script ourselves. The other script that was really important that came out of this paper with Carl was to compare the ribosomes. That's a huge, huge operon that's there. And in that operon is the RNA for both the large, the 23S at the bottom, and the small subunit, which we have at the top. That's the one he used to do the tree, right? And what he did is literally go through and look for signatures. He looked primarily for the red signatures. Those are changes in sequences, something that's there in all the bacteria, but it's a different base or not there at all in the other domains of life. The blue indicates structural signatures, because we now have enough structures we can also see do those areas correspond to regions where you have structural differences. Now, the minute you think about that those signatures are they're idiosyncrasies of RNA or other proteins that are characteristics of the three domains of life. And if you're trying to make a drug to kill off a bacteria, you might think about trying to get a drug that knocks out the signatures in the bacteria, but not in you, right? So here's a real popular place that people look on the bacterial genome to try and knock it out. And then when they talk about bacterial resistance, most of the time they're talking about 2058. It gets methylated, and then their drugs don't work. So like when you take erythromycin, it binds on 2058 on the ribosome of the bacteria. Now, the problem is not all bacteria are bad, right? What is it? I forgot what percentage of the human body is made of bacteria. So when you take antibiotics, you tend to, kill everybody who has the signature there. And so that's why then you have to sort of reestablish the good guys that don't make you sick. But this has been now, these kinds of concepts are really important and for not only for targets, but also in our more recent paper, we were trying to look at how these signatures affect the dynamics of folding and assembly in those, right? So, and you can do this signature analysis with any set of sequences. It can be RNA, it can be proteins. And so you have, this is a standard tool that's in the multi-seq. All right. The other thing that Carl insisted on was flexible grouping of data. After he got the Crayford Nobel Prize, he was asked to give a talk at the U of I. And what he did for the first 15 minutes, he read his rejection letters. It was really sad. So one journal, it was a journal of nucleic acid, say, well, look, we've already published 2 genomic sequences, one for a prokaryote and one for a eukaryote. We're not going to publish a third being prokaryote. And he goes, it's not a prokaryote. Prokaryote's a bad name. It ought to be bacteria, archaea, and eukaryotes. And he got rejected again and again until some colleagues, and particularly the ones that were over in Germany, started to support him because there was other evidence than just doing the signature analysis on the rRNA. The membranes, the morphology of the archaea, their membranes is totally different than from bacteria. So once that finally, they started to support him, and they were fairly influential people, they said, let's let this guy publish it. So he insisted on being able to change the families. Like if I had just prokaryotes and eukaryotes, he goes, take that subheading away and make it, an archaea and a bacteria, right? So he's a firm believer of this because he's even had to change his mind within the classifications of the bacteria and within the classifications within the archaeas themselves. So flexible headings, you can put anything you want there. The other thing that's important is if you're working with biological systems, we do try to make it easy that you can read in the metadata information, and it gets stored. If we go back here and look at this window, there's a little eye over at the end, and you click on that, it brings you into this next window, and that's half the information that you've brought in. So you see the common name, the EC number, some short description, maybe a data source. Carl wanted a place to write notes, but he used to write really nasty things like, this guy gave me the sequence, it stinks, right? And so I'll just put something more benign like, oh, residues were missing, right? But, and then you have to save it, save session, and you can have it next. All right, so just a quick summary. You all know VMD. I think there's a little bit of multi-seq within that, and you know how powerful it is in helping you to model and study your simulations. You can do large systems, do it for longer times. You can do it for coarse grain particles. And then what multi-seq brings to that is now you can start combining that information with genetic and evolutionary information, which is very important. And what we'd like to do is be able to integrate the two. And I'll show you one example of that. Since I don't want to have you be too tired for the next session, I'll just show you a few highlights from that. And then if you're going to do something repeatedly, you really do want to run it in the batch mode or these scripting files. Okay, so that would be the end. We'd have break. I think the break is officially, is it 10:30? So if you just give, well, I'll just take 15 minutes more to show you some of the applications that I promised that we would do here, right? So one of the things is, we'll just skip through this, like now we wanted to study how those guys all work, right? Like how does that guy recognize the anticodon down here, but tell it to charge the amino acid onto the tip of the tRNA way up there? Or how does that guy interact with the tRNA, and how does that change as you go across domains of life? Those are easy questions, so let me just pummel through here. So you would do your MD simulation, you would solvate it, put the ions in, and then you say, oh, I got a question. How does that tRNA communicate with the protein and charge something up in the active site by reading the anticodon down here? So you've already done molecular dynamics study of the recognition of the interface. So what are you going to do with those MD results? One of the things you can do is start looking for correlations in the data. So I'm going to try and see if there's a network of interaction that connects that where the anticodon is with the active site there. So in terms of a network analysis, you have to have a bunch of nodes. And the nodes, they can be like the C alpha atoms in the protein. They can be the phosphate and one in the base for the nucleic acids. So you build a network and you connect it by lines. And those edges, they come from, like we use a simple distance cutoff that says if 75% of the time in the trajectory they are in contact, draw a line. So this is like the physical network. But and you could then ask, okay, how many steps does it take to go from position 1? Maybe that's the anticodon to the tip of the tRNA where it gets charged. But what's better is to weight them by correlations. So when you do the simulations, you certainly can calculate the correlations. And so now you weight these guys. And here's what I mean by the correlations. You just look at how much position I changes with the average over a certain window of time. And then you do the covariance there of I with J. And then you normalize it. And now those links, which were all like just sticks, have different widths to them. So some are more correlated than others. And then if I want to talk about that path, say to go from here up to here, that distance, I just add up those weights, like that 1, 2, 3 through 5 or 10. But now the weights are going to be different. So one path is going to be really short. It'll be the optimal path. And one path will be shorter. So I'm showing you the best one plus a few near optimal ones. You can guess who's the most optimal. And we can let it rotate. And if you were to do a mutation study, this already gives you ideas what you might want to mutate. And not all is lost because there are some other paths that would take you between the two. So this is how we look at information being conveyed. Right? One of the things is when you saw those clusters forming up, some of the other tools that are in these network analysis programs is the ability to say, oh, that's a community. There are more contacts within than between two communities. Now, most of the community software algorithms have come out of social media. So like people studying your cell phone usage, they know with whom do you call a lot and how infrequently do you call maybe into Europe. The same way, like the social media wants to know, are you a member of the Boy Scouts, the Republican Party, and living next door to me or something like that. So what you can do is use this Gurman-Newman algorithm that takes those clusters and divides them into community networks. You see 3 of them here. So now let's apply social networking to looking at the action of charging the amino acid glue onto the tRNA for glue. And so that's the end of the tRNA up at there at A76. As an intermediate step, you have to charge the amino acid with an ATP, make it an adenylate, and now transfer that glutamate onto the OH on position A76. All right, and when you do, things fall apart. You've got it now charged onto the tRNA, and that the AMP falls off and leaves the active site. So how is that done? You've done your simulation of before and after. And now you start looking at those networks for the correlations. And you go, let's look at the 2 just involving that active site. And you see, oh, well, this is a good sign. There seems to be 2 communities, one sort of interacting with the amino acid and one interacting with the AMP. And if you now want to go on and do, say, like a quantum chemistry calculation, and you wanted to split it up into here's the quantum regime, here's the classical regime, these would be some of the atoms that you would want to put into your quantum regime. We tried this, and just to get an idea about what's the barrier for the charging step. Okay, you can also use the information that comes out of the social networking to say, well, how correlated are different parts of this? Can I put a metric on that? And again, you can represent that metric as a tree. So I can first just look at those 2 grouping that splits the bottom part. More or less, this is the anticodon binding domain with the part that's involved in the active site. You go through. Oh, well, only one picture here. It will divide it into each step, and you can think, does that have an evolutionary meaning as you bring in each of those components? So in the network, if you want to do the network tutorial, it does sort of the following things. It looks for optimal pathways. It looks for those critical nodes connecting different communities. And then it starts doing like a highway analysis. What are the paths that are already used the most? And that's called betweenness. So you can get all of that out of there. So those are some of the tutorials that are around this multi-seq and a little bit of the dynamical analysis. But I think you're going to have a lot on network analysis using these Gaussian networks that have been developed here in Pittsburgh. So maybe I think what I'd like to do, oh, I can show you these. Just 1 second before I show the staff here. Just say, I think this is a really great problem. Remember, this was the EFTU that transports the charged tRNA. One's done all the sequence comparison over the 3 domains of life. Look what is conserved. Put in modified bases. There they are. This is, to me, the most interesting region. Some of these modified bases are really critical. That's what keeps the loop open. If you take these away, you get a very short loop, and the only thing that's available is the anticodon domain. And then, if you look at the interactions with the EFTU, the energies are positive that way, negative that way. And what we're showing is the interaction energies between the tRNA and the proteins. And so it sort of makes sense. You know, that phosphate, the RNA is like an electrostatic monster with all those charged phosphates. So that lysines and arginines interact favorably with them is not surprising. But that was just for E. coli. What if I put a mask of sequence conservation over the EFTU? You had to blink, but almost everything stays, right? Now let's look over archaea, bacteria, and eukaryotic systems. And you started to lose some of the interactions. There is a gap that comes in somewhere around 300, but what's interesting is going from bacteria to archaeas and E. coli, they all have exchanged a glutamic acid for an aspartic acid. And people do use this in the design of these non-canonical amino acids and their interactions. And so now, let me go back up. Here's the list of some of those tutorials. We have timings. They take some time. So it's more like you just might want to look through them if there's a particular feature of multi-seq that you want. I also understand people had a little bit of trouble with multi-seq on a Windows machine. That's not our specialty, but we can certainly take a look and give you, see if we can't help you with that. And by we, I mean these guys. So Joe Peterson and Mike Halleck are sitting in the back. Maybe these guys stand up so you can see them. Now, particularly Mike said, yeah, he's a big user of multi-seq, all of a day. So, but he's an incredible computer science background. He's our main programmer for our cell simulations. And sometimes these questions are just a matter of CS or the construct between a Linux machine, a Mac, and this. So he can also help you. Joe is also one of the main developers of, he's a graduate student. Actually, he is an NSF graduate fellow, but he was also in our biophysics program and is really worked a lot on the cell simulations, which we'll talk about after the break. And hopefully you guys will all take the opportunity to try them out, because we've got all of you student accounts for the length of this workshop to run them on our supercomputer in Urbana. It's called Blue Waters. And the main reason we did this is to be able to simulate things in a cell takes a lot longer, right? And if you need, you know, microseconds, milliseconds, then the way that we do it is our program has been developed on GPUs primarily, and that allows us to get to the length scales and time scales that we need. And to have a normal human interact with it, Joe wrote one of the Python interfaces that was given out, I guess, published in Supercomputing 13. So all the exercises he's developed, they're from his paper as well as from Mike's paper, because to make the thing even run faster is our constant goal. And one of the ways to do it is use more GPUs, right? So we have a paper called Multiple GPUs, something, something, something cell simulations, right? And it's a very popular paper, even if I can't remember the title. But he can't either. I asked him yesterday, right? So it's not good. We did a bad job there. Anyhow, so these are the people that developed the multi-seq tutorial, or will help you with it. I should point out Elijah Roberts and John Ergel. If you looked at the bottom of the slides, their names occurred often. Elijah is now an assistant professor at Johns Hopkins, and John Ergel is in a startup company in Texas, making much more money than he would ever make in science. And these are two graduate students. She's now a postdoc that worked mostly on the RNA material that went in there. So they are fortunately not with us today, but as I said, if you have questions about setting up your molecular system and how you can use multi-seq to interpret things, we'll be glad to help you, right? And then otherwise, are there any questions before we have the break? Just about this part? Did I answer enough of your questions about the use of multi-seq? I think he's looking at it as we speak. Okay, so why don't we take a quick break, and then we'll start with the section on the putting all this inside of the cell. We'll do that after the break.